Yeah, I wasn't given a ton of guidance on what to present here, so I've kind of put together a little bit of a mix of things, a couple of vignettes and a little bit of actual of a more science story in addition to just a couple of the things we're working on in microbial community genomics. So I am from the JGI. We are the Joint Genome Institute. We're a sequencing and DNA synthesis center funded by the Department of Energy, and we focus on these three mission areas of the biological environmental research area of the DOE, bioenergy, carbon cycle, and biogeochemistry. We were actually founded as part of the Human Genome Project, but as that research wound down, we became this user facility in support of the DOE mission areas. And so I oversee the Metagenome program, which makes up about 20% or so of the bases that we sequence. Our total capacity this coming year will be more than 100 terabases, which actually isn't that big anymore, but this is pretty much devoted to de novo sequencing for the most part. We don't do any human work, and so we do relatively little resequencing. So this is plenty for us to work with. I also I'm going to give a little bit of history. This is, we've been doing metagenome sequencing since the early 2000s, and although it looks like we weren't doing anything back then, we were actually a high-throughput sequencing facility back then, too. I just like to highlight how much this has changed the field of microbial community genomics. So like I said, I'm just going to give a couple of vignettes on things that we're doing at JGI to try to advance our understanding of microbial communities, including a little bit about long-read data, which will hopefully fit in well with some of the talks we already heard, ways of finding novel sequences in environmental sequence data, and a little bit about genomics of plant-microbe interactions. And so this is, as I said, a little bit of history. This is a slide I made back when I was a postdoc sometime early in 2004 or 5. And this acid mine drainage has already come up, like one of the very first real environmental genomics project that focused on a relatively simple community and was able to reconstruct organism genomes from it. And as it was mentioned, that would really held a unique place in microbial community genomics for a long time. Around the same time, we embarked on a project to sequence soil, and not too surprisingly, really pretty much nothing assembled out of soil. We had maybe 100 megabases of sequence data, which was a lot when it was Sanger data. At the time, we extrapolated and thought we would need gigabases of sequence data in order to be able to assemble anything. That turned out to be a wildly underestimated amount. And in some ways, we even still can sometimes encounter projects where we're really getting mostly unassembled data. However, at the time, we decided, well, let's just do something with those gene fragments, and we did these kind of gene predictions on those gene fragments without regard for what organisms they were coming from, and showed that you could see differential gene abundances in these samples that were wildly different. We pulled in the Sargasso Sea and acid mine drainage, or Sargasso Sea and data from another project. We did do some comparisons with acid mine drainage, not here. And we compared soil to these deep sea samples, and we were able to show, things like genes for harvesting light are in the surface ocean, but they're not in the deep sea, they're not in soil. Things for degrading plant biomass can be found in soil. Genes for osmoregulation are in these seawater samples. And so it was sort of a proof of concept, but I feel like in many ways, ever since then, I've been trying to move beyond this, doing something more than just predicting a lot of gene fragments and comparing the relative gene abundances from sample to sample. But this is, in fact, the way we annotate metagenomes at the JGI is we usually take our shotgun data, we assemble it to whatever degree we can, and then we annotate not just the assembled data, but anything left over that doesn't map to those assembled contigs, and we put it all in that IMGM system that you've already heard about today. What we would like to be doing more and more, and we are working with Jill and others on doing this, is actually extracting genomes from those assemblies and annotating them in IMG. So this is where we figured we'd just partner with the expert, and so we funded work in Jill Banfield's lab on improving automated binning strategies, and also Chongal Pan at Oak Ridge National Lab nearby on just getting better assemblies so that this genome binning actually works. And so, but how are we going to get these good assemblies in order to bin? Because often, even if we have good coverage of organisms, the assemblies can end up being very fragmented. And so one strategy we're doing, in addition to just trying to improve the assembly algorithms, is using long-read data. And I think Jill talked a little bit about Molecular, which has worked great, but is very expensive, and so wasn't a very good solution for kind of converting our metagenome projects. But on the other hand, Pacific Biosciences is a technology we use a lot and has been really revolutionary for microbial genome assemblies. Often the assemblies that we do from PacBio data alone for microbial genomes come out in 1 or 2 contigs, and it's really due to this H-gap assembler that was developed for using PacBio long-read data to assemble microbial genomes. And we still do not do all our microbial genomes on PacBio just because of the cost and throughput, but we are moving in that direction, really trying to scale up our ability to do large numbers of PacBio microbial genome assemblies. And we want to take advantage of this in the metagenome world because honestly, these are pretty much just microbial genomes. They're just all pooled in together. So we just need to figure out how to pull that information back out. This is just a slide from Pacific Biosciences website showing how they've been applying it to increasingly large and complex genomes successfully, and the same has gone on at the JGI. We're doing fungal genomes and plant genomes and increasingly incorporating that data in. Just one little note, on the side, is that actually one of our biggest obstacles to doing this is not informatic or even sequence depth, but it is just getting these libraries in the first place, because the quality of the DNA is critical for PacBio libraries to even get made in the first place. And so you can't get all of this to work if you don't have those big inserts in order to do those long reads on. So you can have a 10 kb read, but if it's just going in circles around a 1 kb fragment, you're never going to get that long-range contiguity information. And we have most of our metagenome samples that we try to submit for PacBio sequencing fail because they're too small to really make those big libraries. And this is just an example of a metagenome PacBio library that actually I do have some data from that looked pretty good, but ultimately the insert size was really right around 1 or 2 kb. We didn't get things that were up in that 10 kb, which would really be great for a metagenome assembly. But even at this length, it has some usefulness because it's still much longer than those 150 base reads. So, we've been trying out PacBio on a variety of samples, but some of the ones, not surprisingly, that it's worked best on are the least complex samples that we have. And so these are samples from a collaborator, Bob Kelly, who was studying not just hot spring samples, but actually like hot spring samples that were enrichments on plant biomass, so specifically cellulolytic organisms from these hot springs. And these tended to have anywhere from sort of 1 to a few dominant genomes. So they were a great test case because we didn't need to generate all that much PacBio data. And we varied from generating just one smart cell of data on one particularly low complexity community to, I think, up to about 17 in a case we were trying to see if we could get multiple genomes out. And these are the results of the assemblies. So if you look at the total assembly size, like how many bases can you assemble in total from the data, you actually do get more total data assembled if you do an Illumina-only strategy, because we had generated a lot more Illumina data than we did PacBio data. But when you look at these are H-gap assemblies of PacBio data. Those are just two different parameter sets because we were just trying this out. The contiguity is vastly better out of those PacBio-only assemblies. We were getting these 2.5 to 3 megabase size contigs, so basically whole genomes from the PacBio-only assemblies of the dominant organisms. And the Illumina-only assemblies, not surprisingly, were much more fragmented, but the hybrid assemblies, too, looked a lot worse than those PacBio-only assemblies. And you have to think about, they have every base of data is in there, but yes, what are you? This is with Spades, which is like one of the only assemblers we've actually gotten to run these kinds of assemblies. Yeah, you give all the reads to the assembler, and it does what it wants to do with the data. There are not all that many assemblers that will actually take both data types as a native data type. But yeah, I do have a slide on working. I think that there are many different ways you could think about combining those data. There are relatively few assemblers that actually will do that for you. But obviously what we want at the JGI is an assembler will do us for us, because we can't have every single project be sort of an R&D effort of figuring out how to get these data to work together. But even in one of these, actually the most simple one, we couldn't even get the hybrid assembly to run. It just, never completed. And that's a common problem that we've found, that even the assemblers that do work with a combination of PacBio and Illumina data will fail on these metagenome samples. If you look at some, I didn't put in more statistics, but even, if you look at the overall, it's not just the largest contig, but the overall, N50, et cetera, you really do get much, especially for the dominant members, you get much bigger contigs out of these PacBio-only assemblies, which tells us that, these hybrid assemblies ought to be able to do at least as well as the assembly with just the PacBio data alone. They have the PacBio data plus all of that other Illumina data, and yet they somehow do worse. So there's a lot of development still to be done. One strategy our collaborator Chongal Pan is working on is trying to improve the way that the data is being mixed, and he is kind of simplest way to think about incorporating PacBio data in with Illumina data is to take the PacBio data, improve its quality with the Illumina data, there's something called polishing, and then assemble those long reads with some assembler that can take long reads. But we've done that, and it doesn't look as good as the HGAP assembler. And we think that one reason may be simply the way you error correct is really important. And in fact, Fungal has revealed a high level of chimerism in these PacBio reads that we didn't really expect, that looks like it may be breaking the assemblies apart. And so he's created a different error correction algorithm that basically uses these PacBio reads just to seed little mini assemblies of the Illumina data. That would cause it to then fall apart if it's a chimeric read, because you won't have any Illumina data verifying that chimeric join. And then you can feed those mini assemblies back into your long read assembly and then get annotation out. And our initial results suggest that you can improve both genome coverage and contiguity, so that we don't have to make this choice, because we were finding, well, we wanted to use PacBio data, but it almost seemed foolish to generate both PacBio data and Illumina data if we knew we couldn't really combine them in an effective way. Or we have to just, annotate both assemblies and give them to the collaborator and say, you'll have to pick whether you want the one with the big contigs or the one with the more coverage, or you can somehow kind of sort it all out. But we want one good assembly, not, not multiple sort of almost good assemblies. So that's what we are working on now, but we are still very optimistic about the PacBio contributions, but we really think this is a space for a lot of algorithmic improvement to still happen. So jump to another thing we're doing in terms of trying to find interesting sequences in our environmental sequence data. This is a project that Adam Rivers in my group is working on, and it started with the observation actually in metatranscriptome data that when we generate metatranscriptome data, often a lot of that metatranscriptome data does not map to the references we want to use, either the isolate organisms or the assembled metagenomes. You find that only a fraction of your metatranscriptome data matches to it. So kind of by necessity, we just de novo assemble those metatranscriptomes and annotate them. But then I found when I was browsing my own metatranscriptome data from wetland samples, as well as other metatranscriptome data that we assembled, sometimes we got these large contigs that were several kb in length, which seemed odd because we knew that those prokaryotic transcripts shouldn't be all that long. And when I went and looked at them, they tended to contain these RNA-dependent RNA polymerase genes, capsid proteins, things that suggested these are just RNA viruses that are assembling completely out of our metatranscriptome data and wouldn't be present in the metagenome data. I don't think that's the only thing that's accounting for the failure of reads to map, but it's one of them. And this is actually just a randomly chosen wetland metatranscriptome data set that I went into the other day and just pulled these out. These are the 3 largest contigs. All of them look like RNA viruses. So I told Adam, we should see how many of these there are. RNA viruses are really poorly represented in sequence databases because they're hard to grow and they have only RNA and people are used to sequencing DNA. And so we have a whole collection of metatranscriptome data we've generated at the JGI from soil and marine samples and other systems, and that we've annotated in IMGM. And we thought, well, let's go see, how many of these viruses we can pull out. And so Adam went in and initially just looked for this RNA-dependent RNA polymerase, which is pretty unique to the RNA viruses, and was able to find over 1,000 contigs larger than 3 kb that harbored this gene. And that is particularly noteworthy when you look at the fact that there's only about 1,400 sequenced RNA viral genomes out there. So we might already have as many RNA viruses in our metatranscriptome data as we have in our reference databases. And these are loosely classified into these various viral classes. I don't know how accurate these family assignments are. They're just based on whatever homology we saw. And interestingly, they are in classes that are not just single-stranded and double-stranded RNA viruses, but also single-stranded and double-stranded DNA viruses. And I think we'd have to, we'll just have to look deeper to see whether those are actually DNA viruses that have this gene or whether the sequence homology is misleading. But Adam wasn't, I thought this was already a pretty cool result, but Adam wasn't satisfied because he felt like we were probably still missing a lot. And so he wanted to go look deeper for more contigs that actually came from these RNA viruses. And one thing he encountered was actually that it looked like these viruses weren't really getting annotated very well in the first place because our annotation pipeline is trained on bacterial and archaeal genomes, not viral genomes. And so he felt like we were probably missing a lot of genes just because you looked at these and you could tell that the annotations were poor. There were very short reading frames or things that just didn't look like they made sense. So we went to all this work to train the gene prediction algorithm on viral data and then go back and try to recall genes and see if he could get better annotations of the viral sequences. But while he was doing that, it occurred to him, well, these gene structures are apparently so different, maybe that's going to be useful in actually finding the viruses in the first place. And so he's proposing to use this gene structure information from the gene callers as another signature to use in the classification of these sequences, which falls in kind of a nice middle ground between searching solely based on homology, which is going to depend on what you have in your reference database, and then searching solely based or classifying solely based on composition, which just doesn't contain really enough information to accurately classify stuff a lot of the time. So he's set up a pipeline where he takes this, what they call these transition probabilities. So it's the probability that a given codon will be followed by a certain particular nucleotide that is a signature feature of these genomes, and then putting that into a machine learning algorithm and classifying. And when he's tested it out on viral and bacterial data, or viral and non-viral sequences just from the reference database, it did a pretty good job of classifying. When you looked at those actual contigs from our metatranscriptomes that harbored these dependent RNA polymerase genes, again, it seemed to do a pretty good job of classifying. And so he is working hard with a developer in our group on developing this into a machine learning classification framework that he's hoping will be actually broader than just finding viral data, but will be able to do a better job of classification by using this gene structure information, possibly in addition to things like composition and homology. And meanwhile, we are also just using this for its initial purpose of searching for more RNA viruses in our metatranscriptome, but we haven't gotten those results yet, despite its attempting to run it all week last week. So finally, a little bit more of a kind of science project. And a long-running effort at the JGI is a study on genomics of plant microbe interactions. And this is a good space for the DOE. They're very interested in applications of plant microbiomes for agriculture. And so we started several years ago a project with Jeff Dangle's lab at the University of North Carolina studying the Arabidopsis microbiome. And this was based on our expectation, basically that the plant microbiome will be just as important in plant health and growth and resistance to disease as the human microbiome is in human health and growth. And so we initially did a large just community profiling effort of greenhouse-grown plants in different soils, of different ages, of different genotypes. The first author is Derek Lundberg, who's here, so you can talk with him. That was published in Nature a few years ago. But based on all of that work, these massive data set, we distilled it down to what we called the Hitlist, which is, again, analogous to the Human Microbiome Project, of reproducibly plant root-associated organisms whose genomes we wanted to learn more about. And we didn't choose to do metagenome sequencing of this endophyte compartment because we found even in order to generate the amplicon data, we had to be creative about suppressing the chloroplast and mitochondrial contamination. And if we did shotgun sequencing, it's just all plant, nuclear or mitochondrial chloroplast. So metagenomics wasn't really a viable first tack, so we thought, well, let's just try to get all of these individual genomes. And our initial strategy for, getting wanted list was our single cell pipeline. We have a great single cell genomics group at the JGI. Tanya Wojcicki has really been the leader in developing this technology at the JGI. And they use flow sorting. They just flow sort based on nucleic acid content. They then amplify and do a 16S sequencing of those single amplified genomes. They call them SAGs. And then we can pick the 16S sequences that we want. So we get lots of different 16S sequences, but then we would compare that to our hit list and say, okay, these things are on our hit list. Let's sequence them. Meanwhile, Jeff's lab said, actually a lot of these organisms aren't really exotic candidate phyla, uncultivable. They're in pretty well-known families of bacteria. Maybe we should just see if we can culture them. And actually, it turns out many of them are culturable. And so they obtained a large set of cultured isolates and did the same thing, match it against our hit list, sequence everything that hits the same 16S sequence, or at least something pretty close to it, and feed it into the genome sequencing pipeline. I'm not going to show data today, but a couple of other things we're doing to try to see if we can capture any more is doing things like briefly cultivating and sequencing the metagenome of everything that grows without actually trying to get it in axenic culture, and also flow sorting the metagenomes. Instead of just flow sorting single cells, flow sorting hundreds of thousands of cells into a well, then you can actually use a harsh lysis, which you think is where we're losing a lot of the single cells, and a non-MDA library prep, just a very low input library prep that still uses PCR but doesn't use that highly biased multiple displacement amplification. So based on this, we've been able to sort of check things off of our list. And the net result of roughly where we are now, we're still sequencing isolates, But this is a tree of all of those 16S genes on our hit list. So every single branch here is a plant-associated organism based on our criteria. And then the boxes are things that we've actually been able to get genomes for. And so if they're green, we were able to get single cells for them. And if they're yellow, we were able to get cultivated isolate genomes from them. And we were really pleased to see that these were highly complementary technologies. We weren't sure how this would end up looking, but in fact, some things were very amenable to cultivation, like the actinobacteria, but totally resistant to single-cell genomics. Again, probably due to lysis, but we don't know, from where we go in to where we get out, we don't really know where we're losing everything. And then things like the beta proteobacteria were great for single cells, but didn't cultivate under the conditions that were used. And then a few things we were able to capture both. And so now we're taking all those genomes and trying to analyze them to understand how these organisms associate with plants. And the initial strategy we've taken is a sort of a comparative genomics approach, taking advantage of all the genomes that are in the database in these phylogenetic groups that we have genomes in that are both associated with plants or not associated with plants. And so we have this list of phylogenetic groups that we had isolates in. Then we went ahead and pulled all the genomes out of the database that were of high quality and could be accurately documented as being either plant-associated or not plant-associated. That was a lot of work in and of itself. We fortunately had some undergrad interns who went into the literature and determined, is this truly isolated from the source it says it's isolated from? And, is that a plant or not? Because of the way that those data are stored and the metadata, are not necessarily machine, extractable. And so they had to look and really validate things that were plant associated. And all in all, we ended up with over 3,000 genomes that we were analyzing, over 14 million genes. This was the effort of Asaf Levy, who's a bioinformatician in Tanya's lab. And he has found some really interesting things, like that when he looked just overall at the features of the genome, they were already at a very broad level, they were different between the plant-associated and non-plant-associated. And so this is showing the relative enrichment of these various cog functional categories in the plant-associated versus non-plant-associated genomes. And so you can look at this fold enrichment. And so in some of these phylogenetic groups, you looked like we had kind of just a general genome expansion. And in fact, in particularly like the rhizobiales, the genomes were actually bigger. You had kind of a bimodal distribution between the plant-associated and the non-plant-associated. So they've obviously acquired a lot of extra genes when they're associated with plants. But in some phylogenetic groups, they actually appeared smaller. There was more of a streamlined genome. So there's not always a consistent strategy for all these groups. When you looked at some of these actual categories, by far the most enriched was this carbohydrate and transport and metabolism one, which we hypothesize is related to the plant to the microbes taking advantage of the carbon sources specifically from the plant. Although I think we probably could have come up with an explanation if it was the opposite result. So I think we need more rigorous testing of those hypotheses. But there were also other, comforting things like secondary metabolites biosynthesis, which we thought would be important. And, a couple other categories that I think we all have to, look at in more detail to see if there's a real signature that we can understand. Asaf also did a much more detailed gene-level analysis and looking specifically for gene families that were enriched in these plant-associated genomes. He clustered the genes a lot of different ways, one of which was just based on raw sequence similarity so that we could capture things that maybe weren't annotated gene clusters, but also the known categories like COGs, KOs, et cetera. And then just looked at how these were distributed between the plant-associated and the non-plant-associated, statistically comparing that to what you'd expect based on the number of plant-associated and non-plant-associated genomes you have. So in this example, for example, gene cluster 1 would seem clearly, statistically overrepresented in the plant-associated. And that came up with a lot of interesting genes in things like chemotaxis and carbohydrate metabolism, secondary metabolite biosynthesis, and some very well-characterized genes like nodulation genes or nitrogen fixation genes that we're sure are involved in plant microbe interactions. Just one quick example, because I don't have much time to go into all the data. This was one gene, operon, that was made up entirely of plant-associated, enriched genes, but also occurred across diverse phylogenetic groups, including rhizobiales, xanthomonaceae, and burkholdeриales. And when we looked at it closely and talked with Natalia Ivanova, who is our all-time reference for all things microbial annotation related, she said, oh yes, that's the biosynthetic gene cluster that's thought to produce gibberellin, a plant hormone. And so this seems to be something that's being picked up by all these different phylogenetic groups and allowing them to communicate with the plant on a hormonal level. Finally, just to kind of take it back to the actual microbial communities we started with, Asaf has looked at the mapping of metagenome reads for the rhizosphere versus bulk soil to these plant-associated genes, and has found also an enrichment for those plant-associated genes in the rhizosphere fraction. As I mentioned, again, we don't really have good endophyte genomes to look at, or endophyte metagenomes to look at, so we're using the rhizosphere here as being sort of a middle ground between the endosphere and the soil. So, I don't know exactly how I'm in time, but when I practiced it, it was barely able to fit in my half an hour. So I want to stress just that I think that these experimental and computational approaches really go hand in hand, because with things like the PacBio, we've really had to go back and forth between getting good PacBio data, figuring out how we want to assemble it, and then going back and figuring out how much data do we need to generate, what size insert libraries, et cetera. So you really need to have these feedbacks, and it's the great thing about being at a place like the JGI that we can do that. We really think the long-read data from PacBio have a great potential for metagenomics, but I think the bottleneck is both the sample preparation, as I mentioned, but also just better hybrid assembly tools, I think, are going to really help in getting these good assemblies. And then we're working on this possibility that gene structure may be useful for sequence classification. This is actually one of the pet interests of Eddie Rubin, our director, is searching for novel forms of life, and he thinks that that anything that can help us discover new genomic space will be of interest in better understanding genomics, and so we're hopeful that this may be something useful in that direction. And then finally, sometimes microbiome research doesn't have to involve metagenome sequence data, and for our plant microbe interactions, we found that these whole genomes really complement the community data in finding these habitat-specific genes. So the PacBio metagenome work was really Alicia Klum as head of our assembly group, and then Chongalpan at Oak Ridge I mentioned, and Bob Kelly who provided those metagenome samples from the Yellowstone hot springs. Adam Rivers, as I said, is leading that machine learning project along with Qingpeng Zhang and Ed Curtin in my group. And then the Rhizosphere project is really driven by Jeff Dangel's lab at University of North Carolina, as I mentioned, Derek Lundberg, and then Sir Herrera Paredes has been super helpful in doing the statistics. And then Asaf Levy and Scott Klingenpeel and Tanya Woika's group have been doing a lot of the work there. And then the last thing is that, as I mentioned, we are a user facility, and we really encourage applications to our community science program. We're well into the current cycle for CFP 16, but we have small-scale calls twice a year and a large-scale call once a year in around March to June. And so if anyone's interested in that, please come talk to me. We also have a joint project with the Environmental Molecular Sciences Lab to use technologies from both user facilities. And yeah, if you want to interested in any of these, just come talk to me, and I can take questions.
Thanks, Susanna, for that lovely talk. So when you scale up from microbial metagenomes to host microbes, sort of hologenomes, will the PacBio technology still get us what we want at the end of the day, or is it just too complex because now you're dealing with all of the host background information as well?
I think it's going to depend a lot on the system you're working in, because I think when we're, and I think it's more of a depth thing than an interpretation thing, because if we're sequencing something like a Arabidopsis, it's very easy to just get rid of all that host data and assemble the microbial data alone because you have the host reference. But getting enough microbial data is super challenging. We've been trying to do transcriptome work in those plant roots and just getting, all plant. So I think that that's more of an obstacle than really any of the data interpretation parts. If you can find ways to enrich for the microbial, I think the PacBio will be useful for a wide variety of projects. And I didn't show, even for complex metagenomes, I think it has its place because it does give you bigger pieces than you even get out of your assemblies often. Because, you assemble a really complex metagenome, most of your contigs are, a couple hundred base pairs, and you can get, good 1.5 or 2 kb or longer fragments out of the PacBio. So I think it will have a broad applicability.
Thanks.
Yeah.
So I have a question about soil metagenomics. When you look at the data, fungal sequences are remarkably underrepresented in these metagenome assemblies. When we go to soil and we use other tools, we find lots of signals for fungi. What are the plans to improve metagenome sequencing so that the fungal component can be better understood?
I've observed this too, that, people expect fungi and then, we get very little. I don't know. I would think that it's probably at the DNA extraction step that you're losing them, not in the analysis downstream. So, I would say if you're after fungi, I think, we have, at the JGI, we have historically a lot of trouble getting good DNA from fungi too, because people have a really hard time extracting high quality DNA from fungi, and you can only assume that that carries over to fungi that are actually in soil. So I think, some of those methods that have been developed specifically for fungi, I would think that, using those in soil are going to get you a lot more fungal material. We actually have someone at PNNL preparing a lot of our fungal DNA rather than leaving it in the hands of collaborators, because it was becoming such a bottleneck, just having the collaborators try to isolate good fungal DNA. So I think that would be the place that I would tackle, more than any of the analysis steps.